for the future ones as well. So I want to quickly introduce our two leaders today. They're both members of TRISEM's advisory board, and they were very kind to agree to lead off the first of these, which we're calling Club EvMed. They're both also from NC State, North Carolina State University. So we have with us Jeff Thorne. He's a professor of biological sciences and statistics at NC State, and his research focuses on developing statistical techniques for analyzing DNA and protein sequence data. And our other leader today is David Rasmussen. He's in the Department of Entomology and Plant Pathology, also at NC State, at North Carolina State University. And his research focuses on phylogenetic methods for tracking the spread of pathogens using genomic sequence data. So I'll turn things over to them, but I again want to encourage people to please participate, by raising your hand or putting questions into the chat function. But we'll let Jeff and David start with a little bit of an introduction to the material they find interesting about this coronavirus.
Okay, well, thank you, Meredith and Charlie. I'm going to try to share my screen with you, so I'm hoping this will work. It worked when we did a trial run. And if it works now, what you should see is a picture of 3 people, and I'm trying to change it to full screen mode. Do you see 3 photos? Does anyone see 3 photos?
Looks good to me.
Yes.
Excellent. Excellent. Thank you. Okay, so what I'm hoping to do here is, first of all, and I will do this even if I didn't try, establish that I'm not an expert on coronavirus evolution, because I'm not. My research area, as Charlie mentioned, has to do with studying molecular evolution. Meredith and Charlie had been really interested in, and I thought it was a really good idea of theirs to have a sort of way to have people interested in evolutionary medicine in the Triangle connect during this time of social distancing. And so we had some email about this, and of course the thing that everybody's thinking about, I think, is coronavirus. So what we thought we would do is just talk a little bit about how coronavirus topics can be integrated into evolutionary medicine courses. And I want to say that I don't have a complete idea as to how to do this. I have some slides to show that hopefully illustrate some things that I think can be conveyed, that I think will be interesting, at least for the next few years, to students about evolutionary medicine and about evolution. In my own situation, I co-teach an evolutionary medicine class at North Carolina State with Marianne Niedzlik-Fever. She was very kind to a few years ago let me help teach that course. And then I want to mention that the points that I do have really aren't coming from me. They're coming from David Rasmussen, the other person who's a co-host here. David is not a coronavirus expert, but he is an expert on using genetic data to study viral epidemics. And the person on the bottom is apparently a character from House of Cards named David Rasmussen. And when I was looking for a picture on the web of the North Carolina State version of David Rasmussen, I saw that there was this character on House of Cards that looks a lot like, I think, David Rasmussen from NC State will look in about 50 years. I thought that the resemblance was uncanny, so I wanted to show that. But anyhow, my my feeling about evolutionary medicine is that one can teach the course in different ways, and I should say that I am learning. I really learned about evolutionary medicine from Charlie Nunn, and in fact, he was kind enough to share lots of the slides that he uses for the undergraduate course that he teaches at Duke, and every one of Charlie's slides is better than every slide that I have ever made. And so for some topics, right now in the class, for example, we're talking about cancer and evolution, I rely very heavily on Charlie's slides. But here we can't blame Charlie. because first of all, you won't see many slides, and second of all, they were really made by me. But so I think a lot of evolutionary medicine courses are aimed at teaching people about different perspectives, evolutionary perspectives for understanding disease, for understanding, why do humans have back problems, for understanding why do we have late onset diseases and so on. In my selfish case, we have at our university, we have no med school, but we have the majority, we have a large number of biology undergraduates who want to go into health-related professions. And I thought that this evolutionary medicine course that I've helped design and that I co-teach, I thought it would be a good way to really trick students into getting interested into learning something about evolution. So a lot of my ulterior motives are really in the evolutionary medicine direction, but it kind of became clear to me after teaching for a while that one way to interest these people who want to go into health-related careers in evolutionary medicine is to think about how can we show them that besides understanding disease, they can actually use evolutionary ideas to potentially treat disease. And so what we usually do is we talk about how we can think about certain topics that are practical and important and applied in an evolutionary context. And I use those as excuses to teach people about evolution, which is molecular evolution, especially because that's what I know the best. So we can talk about how cancer really is evolution of somatic cells. I like to cover genetic pest management. So how for example, we can think about and evaluate strategies that people have for dealing with pathogens or vectors by releasing genetically modified organisms into the environment. HIV has so many great examples of evolution. Influenza does. And then this year it became clear as our pandemic developed that coronavirus is something that's going to be of interest to people for quite a while, and I'm hoping to solicit suggestions after I show you these few slides. And in fact, if you want to talk or if you have comments, additional points, if I make mistakes, you should point those out, because really this should be a discussion. It's just that I'm very good at blathering. And so if people want to inject actual good points, that will be super. There are quite a few resources on evolution with regard to the coronavirus pandemic. My co-teacher, Mary Ann, makes good use of web pages that Berkeley has produced on evolution, and they have a page that I'm sure will itself evolve as our understanding of coronaviruses evolve. They have a page on COVID-19. There's a really nice video. You can see it, for example, on YouTube by Vaughn Cooper from the University of Pittsburgh. And if you go to YouTube and you search for Vaughn Cooper and COVID-19, I think you'll find it. But if you want to look at it and you can't find it, just send one of us email later and we'll let you know. And then I've really been impressed with two websites that David Rasmussen has told me about. And I think many of you are probably very familiar now with the nextstrain.org website that Trevor Bedford's group at Fred Hutchinson in Seattle has put out. And Trevor Bedford is heading the group that realized well before the US federal government realized that there was community spread in Seattle by basically making phylogenies of viral isolates and so on. Their webpage is just tremendous. And then there's also a web page called virological.org where experts on viral evolution will post preprints and we'll have discussions. And for me, as kind of a groupie of viral evolution, it's kind of interesting to go there and to see what people are working on and what their latest work is. David, did you want to say anything about virological.org or other websites that are available?
I think those are the two main ones that I've been following along with the most often. One that isn't evolutionarily focused, but is really interesting is this webpage called COVID Tracker, which gives up to the day reports on the number of cases and the number of total positive versus negative tests done in each state all across the US. So you can get sort of up to the day or even up to the hour updates on the epidemic, although it's not from a molecular epidemiological or genomic perspective. I'll post the link in the comments.
Excellent, thanks. And in fact, I should say, David has been incredibly kind to me to teach me at least something about coronavirus evolution. While he isn't a coronavirus expert, he's one of the developers of these statistical methods for studying epidemics via the genetic data from epidemics. And I've really enjoyed bothering him. I kept it to once a week because I know David has other things to do than deal with a person that's kind of at the interface of, well, it's kind of, you know, caught in between middle age and being an elderly person who comes into his office and just wants to learn stuff. But it's been very interesting to me to get kind of weekly reports from David on his perspective on the coronavirus epidemic. And I think that I did, to my credit, only bother him once a week, pretty much. Maybe I violated that a couple of times. But in the beginning, he was looking at this and he thought the epidemic was interesting, and he was telling me about what people were doing. And it became relatively clear to David by late January that there was, at that time, a high probability that the US was going to experience exactly what it is experiencing. And David made the point also very early on that if we did get community spread in the US, that it was going to be a very different experience for people in the United States than people in China, just because of the way people behave and the way the government is organized. So it's really been interesting to me. And I also realized that, I don't know if he did, but if he wanted to, David could have made a ton of money on investing, because David was well aware of what was likely to happen in the US before Wall Street was.
Several people told me to short the economy, but fortunately, I don't know how to do that type of thing.
Well, he's got a wealth of knowledge, if not a wealth of money.
But it is worth pointing out, Jeff, that you're coming to my office every week in January, but I think almost every week until the very end of January, I was saying, no, I think they're going to get this thing under control. It's a respiratory pathogen with a high transmission rate, but it's not that much more transmissible than flu. So if we put into effect the proper social interventions, we'll get this thing under control. I was very wrong about this. I think
but you were very aware of what was going to happen before the rest of the U.S. society was. You were telling me, and so then I was panicking, and I got accused by relatives of panicking because I was parroting this information that David was getting me. But anyhow, I want to say that the slides that I've produced are really not too much about coronavirus facts. They're more about using coronavirus to teach ideas or statistical methods in evolution. And that's largely because I tend to be fact challenged. And so it's a fancy way of admitting that I'm often fact challenged. But before I go into specific topics regarding using genetic data to understand coronavirus evolution, as David pointed out, it's also interesting to just talk about, and with that COVID tracker site that he mentioned, it's interesting to think about what we're all seeing on from the media. You know, we're hearing a lot about flattening the curve, and as you know, the idea is that for these flattening the curve kind of pictures, on the x-axis we have some measure of time, and on the y-axis we have some measure of the number of people who are going to be in the hospital. And so here's one slide that I picked off the web, and the point of all of these pictures that we've seen is that if we can so-called flatten the curve, then the number of people in the hospital due to this pandemic won't exceed our hospital capacity. So that's been the whole idea, and one of the ways of selling social distancing as it should be. But if you look at this particular slide that I pulled off the web, and then you can look at another slide that I pulled off the web, they're both basically trying to establish the same point. In both cases, we have an x-axis that's time and a y-axis that's the number of cases, and we have some theoretical limit of what the health system can do. So going back to the first picture, if you look at these two pictures, I like one of them more than the other, and this doesn't have anything to do with me having an affiliation or not an affiliation with the University of Michigan. Is it obvious to anyone? I'm not sure how to do it, how to solicit points here from those of you, but does anybody have an idea as to, I mean, comparing these two pictures, one of them to me looks more useful than the other. Does anybody have a suggestion as to whether it's the University of Michigan picture or the Medscape picture?
So I'm not sure if it's related to that point, but it looks like we do have a hand raised.
Oh, great.
I'm gonna unmute that person. Looks like Asprajaya Shankar.
Yeah, thank you for the opportunity. This was in response to Professor Thorne's question. Actually, the Medscape one is more accurate because the healthcare system's capacity limit is much lower. In both the charts, the basic issue is that we are too optimistic about the healthcare system capacity. In reality, it is far, far lower. It's more closer towards the x-axis than it's being shown now. Thank you.
Thank you. I think that's a great point to make. Another point that occurs to me is if you look under the area of these two curves, if we do take social measures, the one that has the lower peak, and if we don't, the one that has the higher peak. In the first case, just by eyeballing, it looks like the area under these two curves, which somehow represents, is going to be very correlated with the number of people who get sick with the virus. The area isn't too different in the first case, but in the second case, you'll see that if we take the protective measures, it's not just the situation of the healthcare system being overwhelmed or not being overwhelmed, but also we have to consider that if we can have an effective social distancing policy for long enough, fewer people will just get infected. And I think the second picture more accurately conveys that. So I feel like some of the pictures that we're seeing and so on are maybe sending not quite the right message, but anyhow, and you can find pictures that are certainly more nuanced than the ones that I've shown you. Anyhow, what I wanted to get onto is just some ideas in evolution that we can use the coronavirus epidemic to convey to students, and some features of the coronavirus that themselves, this epidemic, that can be illustrated with the pandemic. So an obvious one is we can talk about phylogenies, we can talk about evolutionary trees, we can get genetic data from a bunch of viral genomes, we can make an evolutionary tree, in this page from Berkeley that I gave you a link to. They've done that, and what we see is the basic scheme of what right now people, as far as I know, have found. It may be that there are newer results that some of you are aware of, and I'm keen to learn about newer results, but basically the picture is, and this shouldn't be just viral isolates from people in China, this should be viral isolates from people who've been infected around the world. It looks like there's a single most recent common ancestor of the people who are infected that's more recent than any of the coronavirus family members that have been isolated from other non-human organisms. There's a particular isolate of the coronavirus from a bat that on a genome-wide level looks to be the closest relative to, according to the phylogeny, to the viruses that are causing the epidemic. However, it's also the case that coronaviruses isolated from pangolins are at a genome-wide level pretty similar to what we're seeing in the epidemic now. And then other coronaviruses estimated from bats are a bit more distant. And so there's been a lot of interest, and this is I think, David can comment on this, but I think this is a really active research topic right now as to exactly what can we learn and what should we know about how the cross-species transmission event happened. So one question is, we can look at, with phylogenies, we can look at the transfer history, we can see which sequences are closer or not, and if you're somebody who wants to convey how we just understand evolution using DNA sequences, you can use this potentially as an excuse to teach about phylogenetics, which I'm very happy to do. David, did you want to say anything about that topic?
Well, I guess one sort of interesting topic for discussion is that tree of evolutionary relationships among the SARS-CoV-2 human samples and the samples from bats and pangolins is probably going to change a lot over time as we continue to sample from different non-human hosts, including more and more bat populations and more pangolin populations. So I don't know, how would you explain to your evolutionary medicine class that, you know, as new data comes in, we're constantly going to be changing our theories about what is the proximal origin of this virus in these non-human populations?
I think that you just explained it really well. I think and it might be, so for me, the next time I'm teaching this, co-teaching this class right now, and I won't be co-teaching evolutionary medicine again until the spring semester of next year. And it might be that the sampling of, probably it will be the case, that the sampling of these coronavirus family members from other non-human species will be much, much more thorough then. And so we might have a much better idea of what happened by then. But I think, you know, it's always good. I don't always do it effectively, but I think it's always good to explain not just what we know about science, but things that remain unknown, things that science hasn't solved or in some cases can't solve. But I wanted to say that another thing that we can do is we can estimate the times of these events. We can talk about molecular clocks. We can, for example, use this great website that I mentioned, nextstrain.org. Nextstrain, one thing it does is it has for the COVID-19 pandemic, it has for flu, it used to be called nextflu.org. It has for Ebola virus. It has these different ways of visualizing the data. And in this particular picture, which is a screenshot taken from nextstrain.org, what we're seeing is the date at which viral isolates were obtained on the x-axis, and number of substitutions from the number of nucleotide changes from the earliest genetic isolates, the earliest isolates, is basically shown on the y-axis. And so the higher the numbers are on this picture, the more diverged they are from these ancestral, well, maybe not ancestral, but from these early isolated sequences. And the thing that David pointed out to me early on in January is that at the time, if you look at some of these early January amounts of genetic variation, they're very small, which suggests either that COVID-19 is not evolving very quickly, and it actually is evolving moderately quickly, maybe not as quickly as, say, HIV would within a person, but it's evolving, it's a retrovirus, it's evolving pretty quickly. But the reason there wasn't much genetic variation is presumably because these different isolates from humans all had a recent common ancestral sequence, and so there wasn't much difference between them. And that posed problems for the approaches that David develops for understanding epidemiology. We'll talk about them in more detail for understanding epidemiology from genetic data. If you don't have much genetic variation, then it's, there's a lot of ambiguity in the evolutionary tree. And when there's ambiguity in the evolutionary tree, it's hard to learn much about the epidemiological parameters that we'll talk about that might be interesting. But what you'll see, and what David has pointed out to me subsequently, is now the amount of genetic variation is substantially more that's circulating within humans. That means that there's more structure to the evolutionary tree. And as we'll see, that means that we can potentially estimate epidemiological parameters better from genetic data. But first of all, we can talk about how, instead of using fossils to estimate times since common ancestry, using fossils plus sequence data, the idea is sequence data tells us amounts of change, but if we have a fossil, the amount depends on the product of rate and time. And if we know a fossil, then we can estimate the amounts of change from sequences. And with fossils, because we have time points, we can disentangle the rates and times. And what people recognized quite a while ago now in viral evolution is that they have a substitute for the kinds of paleontological evidence that people have used for studying long ago molecular evolution. They have times of viral isolation. And when you're looking at HIV or flu or coronavirus, because those genomes evolved so quickly, there's information in available in when was the sequence isolated. But and we'll see a picture about that before. Another thing that you can see from the nextstrain.org tree is that different viral isolates are colored differently, and they're colored according to where was the geographic origin of the isolate. And so Trevor Bedford's group has impressively been able to figure out when local areas have been separately colonized by using the tree, have been separately colonized by multiple viruses that have been migrated into that area, or whether they've been colonized by just one. And so there's lots of work on viral biogeography, trying to understand migration events and so on, that I think is continuing to improve, and that we have the potential to try to convey. At the beginning of this semester, when I showed David the syllabus that I had, this was before we were as focused on the coronavirus as society is now, I asked him what topics should I include, and he said, well, one topic that you should include in your class, which is really important, and he was very polite, but he was it was clear that he thought, probably accurately, that I was an idiot for not having that topic, was evolution of virulence. And that's something that I haven't taken David's excellent advice on, but now I'm even more interested in it because, as you know, people are very concerned with how will these coronavirus genomes evolve over time. Will they become more nasty? This will be especially important once, if ever, we get presuming we get a vaccine. How will the natural selection on these coronavirus genomes affect what happens with virulence? But anyhow, so that's an area of theory that I want to learn more about, but that I really don't know so much about. David, you should jump in here because you actually know that theory.
Yeah, the evolution of virulence stuff is fascinating because it goes into these really interesting trade-offs as well. Trade-offs versus in the transmissibility of these viruses versus how much damage they actually cause to their hosts. So the idea would be that there's some intermediate optimal amount of damage that the virus would cause that would maximize the virus's potential to transmit, in that people wouldn't be so sick that they would need to be hospitalized or lying around in bed, but they could still be sort of up and active as people commonly are when they have the common cold. But then also at the same time, you want to optimize actually transmission potential, which generally requires selection for higher and higher within host viral load to get viral load up. So viral titers are high enough that even small droplets in our phlegm or whatever comes out when we sneeze actually contain enough individual viral particles that the virus is capable of transmitting. And so it's also couples very well with talking about epidemiology as well. So in epidemiology, we talk a lot about R0, the basic reproductive number, which is the number of new cases and an average infected individual contributes to the next generation of cases. And what you can show using pretty simple evolutionary epidemiological models is that you will actually get selection for the virus to maximize its R0. So not necessarily to maximize within host viral loads or virulence, but to actually balance these two things so you actually get an optimal R0 or basic reproductive number. So the virus actually, there is selection on the virus to actually increase its fitness in terms of the number of infecteds in the next generation. If there's time now, we might want to circle back to the mutation rate stuff because there's a bunch of questions in the comments while you're talking, Jeff.
Oh, sorry. Well, and I saw somebody had raised their hand too. When I'm talking, I'm not seeing that. So why don't we do that?
Let's get to the hand raise first, and then we'll circle back to the chat box. So Dan Eisenberg, I'm going to unmute you now. Go ahead, Dan.
So I wanted to follow up on David's point about the evolution of virulence. It occurs to me that maybe one prediction from that, as well as other evolutionary perspectives, would be that you'd have more virulent forms coming in hospitals from patients who are more immobilized. And I've heard that anecdotally, but I don't know if anyone else has heard if we're getting good evidence for that. But that would seem to be a way to link in the evolution of virulence more.
Yeah, that sort of reminds me about this classic sort of antidote that's in the literature. Maybe I shouldn't call it an antidote, maybe it is empirically supported, but for a lot of these waterborne pathogens like cholera, you could actually have the virus be quite virulent and immobilize people who would just be, laid up in the hospital bed because with these waterborne pathogens, someone can obviously be sick in bed, but their diarrhea still can get into the water and contaminate and make other people sick. So if transmissibility becomes uncoupled from the mobility of the host, then it is possible to get almost like this runaway selection for increased virulence because there's no reason why the host has to be sort of up and mobile to spread the disease.
Yeah, so David, it looks like in the chat box, you kind of addressed this a little bit, but we have some questions about what flattening the curve will actually do to the virulence. Looks like Byron Adams had said another benefit of flattening the curve and reducing transmission is that it should drive the evolution of reduced virulence. But then others have pointed out that it might encourage strains that are even more virulent because only the strains which survive are those that can replicate during social distancing. Yeah, what are your thoughts on flattening the curve and its effect on virulence?
Yeah, so I don't know what will actually happen with SARS-CoV-2. It's unclear if the virus actually has any genetic variability in its population for selection to act on in terms of these virulence traits. But what I would expect is if we're actually all at home distancing ourselves, there would be, I guess, selection on the virus to extend its effectively its generation time, how long people stay infected for. So even if they do a really good job of staying home for the first week or two that they're infected, they're still infectious in week 3 when they finally have to go out for groceries. And there might also be selection for decreased symptoms too. It might be that there's selection acting on the virus to be more stealthy. So people are actively transmitting even though they don't know they're sick. The big question here is if, as I said, if there's actually any genetic variability in the viral population for these traits. And I don't think we know that, but what we do know, and it's kind of interesting, is that we do know there's a lot of genetic variability in mammalian populations for how we react to these viruses. So Ralph Barrick's group at UNC, for example, has done these tremendous genetic studies in lab mice where he's shown that there's tremendous genetic variation for how aggressive the immune response is in reaction to infection by coronaviruses. And sometimes in some mice seem to be basically genetically predisposed to have an overstimulated response that basically kills the mice, whereas other mice seem to be genetically predisposed to basically tolerate infection.
So I was just scanning through some of the comments in the chat that were made, but I think if anybody wants to correct me or raise a question for David or whatever, this is a good time. Otherwise, I can just continue with the slides that I had. Okay, I'll start sharing the screen again, but I haven't figured out how to be smart enough to see the comments while I'm talking, so I apologize. So if there's a big error that I make or a good point, any point that people want to make, just let me know. One thing that I think is interesting in general in evolutionary medicine is to talk about and to think about, and I can't claim to have a good idea as to what to do, but to think about in the future, if there is bioterrorism because of a genetically engineered pathogen such as a virus being released, can we detect it? So I think you're probably all aware that there are US politicians who've accused the Chinese government of engineering this virus with very little evidence. And there are Chinese government officials who have accused the US of basically releasing this virus, again, with very little evidence. And although there isn't any evidence, I think the question is really interesting. How would we decide whether COVID-19 is genetically engineered? There was a recent paper in Nature Medicine by Anderson and colleagues, and they made a few points about why the genetic data don't support the hypothesis that the COVID-19 sequences, our genomes are genetically engineered. One point that was made is that the particular receptor binding domain that the virus uses to get entry via the ACE receptor in human cells, the virus uses to get entry into cells, that particular region of the genome, people had previously identified that there were 6 key amino acids in this receptor binding domain. And what they see, which is interesting by itself, is that basically all of those 6 key residues from some pangolin isolates basically have all of the 6 positions in the protein basically have the amino acid, as I understand it, that people thought would be ideal for letting a virus into the cells. Whereas although the pangolin sequences are really similar in this receptor binding domain to kind of what people thought might be optimal, the viral isolates from bats in general are far more similar to, or at least the one viral isolate, this RATG13, is far more similar to the COVID-19 sequences. And that suggests that maybe if you just think about that and you don't think too carefully, you say, oh, well, maybe there was genetic engineering because there's mix and match between the part that works from pangolins and the bat sequence that in general is similar to the viral genomes. However, it's the case, as I understand it, that the RNA binding domain in general it's got a way of in COVID-19 of binding to the ACE receptor that's different in some ways, but effective, different in some ways than people had thought would work. And so evolution finds its own solutions that maybe humans weren't aware of. And it's also the case that the COVID-19 genome isn't similar to the coronavirus genomes that have been studied heavily in molecular genetics. One might imagine that if bioterrorists were going to engineer something, they would engineer something using genomes that had already been studied and then tinker with those genomes, which hasn't been the case. But in general, I think based on at least molecular phylogenetics, maybe people are aware of approaches for assessing this. But I think in general, molecular phylogenetics needs to have more approaches for seeing whether possible sequences really have a phylogenetic structure, or in the complicated real world case, we have to think about phylogenies and we have to think about recombination. I think it's interesting for techniques to be developed that will in the future try to identify engineered sequences. I'm just not quite sure how that will come about. So anyhow, to me, that's interesting because it is a little bit hard to separate. We know that apparently, or experts know that coronavirus sequences have a lot of recombination. And what that means is that if you get a bunch of coronavirus isolates, different regions of the coronavirus genome will have different evolutionary histories. And people have thought, really, really smart people have thought for a long time about how to integrate recombination and evolutionary trees, but it's still a really challenging area. And the caveat of a lot of approaches that we have for analyzing genetic data, whether it's from viruses or from other natural other organisms, the techniques for dealing with recombination still need, I think, a lot of work. Does anybody have any comments or questions before I start talking about, in the last few minutes, a little bit about epidemiology? Okay, so David mentioned this R0 number that people are very interested in. Epidemiologists like to estimate this with a pathogen. There are classical epidemiological methods that don't use genetic data to estimate these numbers, and those, my understanding are still the state-of-the-art kinds of approaches. But David Rasmussen, his postdoctoral advisor, Tanya Stadler, and quite a few other people have developed techniques that are starting to be able to exploit the information from genetic data to estimate these things. And as David, I think, mentioned, if R naught is less than 1, then you don't expect the epidemic to spread. And if R naught is greater than 1, you expect the epidemic to spread. And then there are rules of thumb that will depend a lot on details, but in a very crude way, knowing the value of R naught tells you, if you had a vaccine, what proportion of people would you vaccinate to stop the epidemic. People have estimated R naught for different sorts of viruses. And so at the bottom, you see some estimates that people have obtained. What the estimates are, my impression is, of the R naught values for coronavirus they're still a bit crude, but they're between 2 and 4. And one thing that I think was long clear to experts in epidemiology, but it has just become clear to me reading these papers about coronavirus, is I always thought R naught was a property of, I guess I never thought about it, but I thought R naught was a property of the pathogen. And in reality, it's an interaction between the pathogen and society. So if you have a society that's practicing social distancing, then basically those social practices decrease the value of R naught. And the big hope is that if we can get R naught to be small enough, then fewer people will get infected. So I thought what I would do now is just give a brief picture of molecular epidemiology kinds of models, first connecting them to classical epidemiology models. And the most basic sort of model, as has been pointed out, I think, in the chat, these models have been improved in lots of ways, and there are really smart epidemiologists who know the strengths and weaknesses of these models, and they have some models, but they're really simple models. The SIR models that we can teach our undergraduates about say that each person is going to be in one compartment. In the SIR models, the 3 compartments are the susceptible people, the infected people, and then the recovered people who might be, after they're recovered, they might be immune to getting infected again. And one way to study SIR models is to have a bunch of differential equations. You can say, well, susceptible people can get infected, and so the change over time in the number or the proportion of susceptible people, it will be negative, and it'll depend on how many susceptible people we have, that's S, or the number, the proportion, how many infected people we have, because the idea is it's interactions between susceptible and infected people that cause infections. And then beta is some term that represents, for every time we have an interaction, if you think of susceptible people bumping into infected people, there's some chance that a person who is susceptible gets infected, and then that susceptible person goes into this compartment. Infected people might recover with some rate, and here's the term that explains why more people get added from the susceptible part. And anyhow, we have these differential equations, and we can estimate the relative rates of them. And it's, once we know for a certain compartmental model, such as SIR model, once we have estimates of the parameter values, then we can make public health decisions if we want, using those parameter estimates to derive something like this basic reproduction number. So the idea in molecular epidemiology is you get genetic data from your viral isolates. That gives you a tree. One point is that once you've inferred that tree, different isolates have different times. So here's an isolate that happened earlier than these isolates. And because you have different times, so-called serially sampled data, what you might imagine is that, for example, this isolate is closer to the root sequence than the isolates that were obtained more recently. And that's what you expect because of the differences in sampling times. And because you expect different amounts of evolution when there have been different time durations. For example, since the common ancestor, you can estimate rates not from fossils, but from different sampling times. So in the molecular epidemiology literature, the idea is genetic data tell you about trees, and the trees show you about where lineages come together, where they coalesce within the viral population. So for example, maybe some virus infected somebody and that person infected people who led to two different lineages here. And if you think about coalescences, the idea is that this pattern of coalescent events on the tree tells you something about the number of infected people at each point in time. And I'll show you a picture to illustrate that. But the point is, when you can estimate the number of people who were infected at different points in time during a pandemic, that tells you something about these parameters, because basically the values of the number of infected people will influence the behavior of the epidemic. And here's the idea. One idea of molecular epidemiology. So you have a bunch of viruses. Maybe this is, the viruses from the US now. You isolate a couple of the viruses, and then you think about when did the most recent common ancestor of these two viral isolates exist? It might have existed pretty recently when the population was moderately big, or it might have existed long ago. And the idea of these kinds of approaches is coalescence is more likely to happen one generation ago if the population size is small. Because if the population size is small and you have two viruses and you're saying, did they coalesce? Did they have a most recent common ancestor in the previous generation? Then the very most simple idea would be that if you have two isolates, the chance that they both have the same parent, and lots of assumptions would be made here, but the chance that they both have the same parental genome would be 1 over the number of isolates they have in a certain amount of time. And so coalescent rates per generation are higher when populations are small, and the chance that in a large population there's a coalescence, the chance of a coalescence event is small when the population is large. And so basically, if you have lots of isolates, if you have lots of genetic data so you can characterize these evolutionary trees, you can see the relative rate of coalescences at different times here. And if you can calculate these, then you can estimate the population sizes at different times. And you have to take into account the fact that with these compartmental models, the rate at which each infected person passes on the virus to another infected person, that really actually depends on the values of the number of people who are susceptible and infected. So basically, in these epidemics, a generation is kind of like, how long does it take for one person to be infected by another? And what I want to say is, to me, this area, even though I'm not an expert on it, this area is something that I want to learn how to teach. And clearly I need to learn. I just think this is a really cool area. And I guess I'll stop there and see if there are any comments. I'll stop sharing my screen too, maybe, huh? David, what did you want to add about things that I screwed up?
I think you did a great job, Jeff. I have nothing to add on the Osiris stuff. And I guess what's neat is that we'll be able to do this in real time as more and more sequence data accumulates. And actually, now it's even possible to do these sorts of exercises in a class with your students. You can, use coronavirus data to, estimate molecular clock rates, for example, either using the root to tip sort of regression approaches that Jeff was showing, earlier, or in phylogenetic software packages like Beast or RevBase. So I think there's a lot of opportunity for both the molecular epidemiology and the molecular evolution to get in the classroom now.
So I don't have much else to say, but I'd really appreciate any ideas that people have for me, selfishly, about what can be integrated into an evolutionary medicine course. I'm, as maybe we all are, I'm just really keen on knowing more about coronavirus. And so I really appreciate all of your being here. I'm going to read through these comments later because it looks like there's a ton that I should learn. And maybe this will motivate Meredith and Charlie for figuring out what to do for future iterations of this.
Looks like we do have a hand raised from Mark Friedman. I'm going to go ahead and unmute. Mark, go ahead.
Hello, and hello to all of you, and thank you, Charlie, for organizing this. I sort of fall outside the curve of all of you. I am a high school physioanatomy teacher who has worked with Randy and Barbara to develop high school resources on evolutionary medicine and then get them into some of the schools out here in Arizona and other places. In other words, making them more understandable to high school seniors taking physioanatomy, AP biology, etc. On a high school level, giving them a step up as they enter college, and many of them consider a field in medicine. So taking the EvMed resources. We have had requests and been able to get some new material on viruses through Carla Moeller onto Ask a Biologist website. We have requests from the LA Unified School District of tens of thousands of teachers to make more science material such as this available online. Right now in high schools, many schools are studying units in genetics and the immune system, which is perfect. I think that if we can develop, and I am willing to help on this, a 10 to 15 minute video, PowerPoint with narration or notes that we could then put up on the EvMed website, and then we can send that out to thousands of teachers, that they now have this resource which will include, not to use pejorative words, sort of a dumbed down version of what we've been discussing today, but a more understandable, but at the same time keeping the high level of science, talking about the immune system, mutations, R0, etc. All of that is fine. In ending, I want to say that if anybody has any information on the use of interferon alpha 2b, which was developed in Cuba, is being widely used in China by the millions of doses, that has been shown to reduce the symptoms, the duration, and the morbidity of this. We are putting together that scientific information, scientific papers. We already have some to advocate breaking the US blockade and allowing this medicine in from Cuba or China. So if anybody has that information or would like to help on that campaign, I appreciate it very much.
Thank you. I don't know anything about the latter issue, and you clearly know a lot about the former issue. I think your suggestion to make some kind of accessible resource for people is just tremendous, and I am happy to help in any way that I can, although it's probably smart to get the experts on coronavirus and experts on these methods like David to to be the key players. But I think that would be great. I have been thinking about my class is in kind of an upper division class at an undergraduate level, but I think it's tremendous to think about how some of these ideas can be conveyed to other audiences.
Yeah, so real quickly, it looks like we have a question from Emily Webb in the chat box that fits in nicely here. Sounds like a lot of these recommendations were for an upper level course. What suggestions do we have for core ideas that you could teach for lower level courses like evolution genetics or even an introductory biology course? And then we'll jump to Jay, who has a hand raised, and go from there.
So I think that the R0 thing, one could develop simulations to show what happens to an epidemic for different values of R0, and it would be important to show the stochasticity, to show that just because people are lucky or unlucky, in one case a virus might turn into a pandemic, and in other cases it might not. I do have feelings for how we can talk about phylogenies, but I think lots of other smarter people have done a really good job of teaching about evolutionary trees, which is integral to molecular epidemiology. And also I think the divergence time topic can be conveyed pretty easily to people. But as far as the molecular epidemiology beyond the R0, I don't know. What do you think, David?
Yeah, I think there's a lot that I can actually be taught at a high school level. I feel like, unfortunately, I might be the wrong person to ask about this because, like Jeff, I teach a graduate level molecular epidemiology class here at NC State. So we actually talk a lot about how to, the statistical methods for actually fitting these types of models to data. But I think there's concepts that absolutely can be taught at a high school level. For example, the molecular clock rate. The molecular clock rate ultimately boils down to the same types of high school physics problems that we think about all the time. I mean, the amount of substitutions that a virus is accumulating is basically equal to the mutation rate times the amount of time two viruses have been separated from one another. So it's the same thing as sort of the classic high school physics formula for calculating, the distance that a car has traveled. It's rate times time. So there's definitely very simple, easy ways of taking these concepts from molecular evolution and tying them back to things that high school students are already familiar with because they already know about things like rates and distances. And certainly if you have, a high school class that, where students are maybe taking something like AP calculus or something like that already, then you could even actually go into the SIR model equations, because all those equations are derivatives, and they're a great example of how a derivative is used every day in modern science. And we calculate rates of change in terms of the number of infections or susceptibles in terms of derivatives in these models. So yeah, if you're a high school math teacher, my God, there's so much you could do with these types of models. But it's been a long time since I've been in a high school classroom. So I don't know, maybe a lot of these sort of scientific applications of calculus are already being taught.
Great. Let's go to Jay Labov. I'm gonna unmute Jay now with a question or a comment. Go ahead, Jay.
Muted me again. There you go.
Oh, sorry.
I actually have two very different comments. The first one relates to teaching in high schools. And I think one of the issues here is not only what's taught, but how it's taught. And so it turns out Dan Grunspan from Arizona State University and I actually did a session before the exponential growth in Virginia at a Virginia high school where there were STEM students from 9th grade through 12th grade. He had participated virtually, and I was there in person. And what we did was a whole session. We were going to get to evolutionary medicine of COVID-19. And we had them doing a lot of active learning and participation. So for example, one of the things we did was to have them role play. What are the questions you would need to ask based upon the group that you're in as to whether the county schools that they're associated with, Fairfax County, the largest one in Virginia, should close or not as a result of COVID? And so we had some people representing students, others representing parents, others representing the superintendent's office, others representing the media. And we had this incredibly rich discussion, which gets them then thinking about all of these different areas, you know, and how they relate to the science. It prepares people to think about the science. Then Dan, mostly with me interjecting, we did a number of parts of trying to help explain the kinds of things that were talked about here at a level that high school students would be able to understand. We interspersed that talking with a number of different exercises. For example, not just telling them about R0, but getting them to calculate R0 and what the implications are for that sort of thing. I've been talking with Dan after this because it turns out we did an 1.5-hour session. And then we actually ended up doing close to another 1.5-hour session with people who were interested and who came back after they had to go to another class. And they were asking incredible questions. And so I don't know whether all of you would be interested, but I'd been talking to Dan about the idea of possibly in Athens, which obviously is moot now, doing a workshop on how to actually teach these sorts of things and using what we did at Edison High School in Alexandria, Virginia, to kind of let everybody participate virtually. In other words, having people pretend they're students and then just kind of working through this and then asking people, so what would you do better? How do you see this relating to your class? How does this relate to undergraduates and graduate students? So I can't speak for Dan because we haven't gotten that far yet, but I personally think it would be an interesting kind of exercise to do, and how we do it online would actually be interesting also, because everybody has to teach online now. So it might also give us some additional experience about that. I should also say that for those of you who don't know me, I am the current chair of ISEM's Committee on Education and Outreach. So I'm thinking about these kinds of things all the time. And I spent 24 years working at the National Academy of Sciences, Engineering, and Medicine on how to improve STEM education. The second thing, and I don't mean to sound glib about this, but Winston Churchill, I think, was the first person who said, never let a good crisis go to waste. And what I'm wondering is when we hear people on TV talking about the epidemic, the pandemic, there is no evolutionary perspective in that at all. And I'm wondering whether we might have any opportunity, at least in the United States, and I don't know who the counterparts are in other countries, to talk to people like Anthony Fauci and Deborah Birx at the White House and help them better understand the complexities of all this, if they don't already, based upon the ideas of evolutionary medicine. I'm not sure who would do it. Maybe ISEM could figure out the group of people who might then be able to make those kinds of contacts, possibly through senators or representatives to try to get to them. But I'd been talking a number of times to a person at the National Institutes of Health who basically said he's been trying for a long time but can't get his colleagues interested in seeing the value of evolutionary medicine to the kind of work that they're doing. And it just seems to me now that we may have an opportunity here because people are focused and they're looking for all kinds of ways that they might be able to approach this from different perspectives. And I'm just wondering how evolutionary medicine or the things that are included in this might be a perspective that could be brought to the experts, which ultimately, if they see the value, may allow them to think about maybe we ought to be funding something like this to a much greater extent. And I'll stop there. Thanks.
So
okay,
David,
thanks.
oh, go ahead.
Go ahead, Jeff.
So David probably has much more well, I know he has a more informed perspective than I do, but this Trevor Bedford from Fred Hutch, who's written this who's headed the team that's made the nextstrain.org site, he really was the person who showed via, I think, evolutionary trees that there was community spread in Seattle. And so he's gotten attention from the press, but I'm not sure that the fact that he used evolution to show that there was community spread has been effectively communicated. And I would think that his work would be, and his direction would be one way to persuade people of the value of evolution.
Yeah, that's a great point, Jeff. Really, really fantastic. And your points as well, Jay. I think we should really follow up on some of those. We're going to close down the discussion though, because we have a number of people who have to leave, and we've already had a few people leave. But I just wanted to thank you, Jeff, and thank you, David, very much for taking the time out of your schedules to have another Zoom call and even to lead one with such a big group of people. This was fantastic, a great introduction to different things that can be brought into the classroom. And I learned a lot in the process. I also learned a lot from just looking at the questions that came in. I mean, things I'd never even thought about. And I hope we can somehow archive that or record that as part of the record of this. But thank you guys so much for participating and leading the first one of these Club EvMeds. And I also want to thank Meredith as well, and thank all of you for attending. I'm really excited about this, and we have a few ideas for the next ones. And so please stay tuned. And if you have ideas, let us know. We're going to stick with the theme of SARS-CoV-2 for a little bit, and probe different aspects of that. This first one on teaching evolutionary medicine, but there are a lot of other things that we could talk about and sort of dive into in more detail. So stay tuned for that, and thank you all very much for joining. Really great to see you here.
Charlie.
Thanks, David.
Thank you all for doing this.
Thanks, Mark. Great to see you.
Yes, good to see you.